Okay, so welcome to lesson 2. We're going to talk about muscle contraction. So this can be a little, I don't know, intimidating but once you get through it, it'll make a lot of sense. Okay, and we'll go over this in class as well. So there's only two types of cells in the human body that have excitability. One is the nerve cells, and the other is the muscle cells. They're very interconnected. So in order to have contraction of a muscle, you have to have stimulation. So you have myoneural stimulation. That simply means muscle nerve stimulation. The stimulation by the nerve impulses are what causes the contraction of the muscle. There's muscle proteins that have to move against each other, and that's what causes the muscles to contract. But it has to start with a impulse in order to start that contraction. Now there's motor units that are consist of nerve cells and all of the fibers that they stimulate. A single um, a single stimulation between a muscle contraction, a single impulse to a single muscle contraction is a twitch. Okay, uh, when you have movement, you have sustain. There's more than one contraction. Um, so you have this motor neuron, and that stimulates all of the the skeletal muscles in that motor unit. So there's a junction between the motor neurons and the nerve cell that is stimulating it. It's like a space. It's a gap that the magic happens in. That there's um, neurotransmitter gets secreted by the neuron into that uh, synaptic cleft, and that's what stimulates the membrane of a muscle cell or the sarcolemma. All right. So contraction of muscle proteins starts with action potential and it requires energy. So you have to have ATP before anything can even happen. And the strength of the contraction depends on the duration of the stimulus, the speed of application, the weight of the load, and the temperature. So I can tell you, if you ever, one of the most sustained one is childbirth, labor. If you ever see a woman who is in labor, she is very hot, she's sweaty, she is working really hard. And those contractions come one after the other, and they are sustained. So you can literally see the contraction. It starts, it goes up, it's like a bell curve, and then it comes down. And then the time in between the contractions speeds up. So you may start your contractions 2 minutes apart. By the time you're in active labor, they may be 30 seconds apart. They are sustained, fast, and they are very strong. There's a lot of energy um, that is produced. Your body is tense, and there's the temperature rises. So that's just an exact example. So let's see what happens. Okay, so you have a resting cell. So you have action potential. Okay. So what happens is there is a stimulus by a nerve. Okay. Now, you touch a hot stove. That sensory goes to your brain, and your brain sends a message back that says, move your hand, stupid, because that's really hot. So you have that stimulus. Now you have your action potential. Um, that nerve stimulus um, deposits acetylcholine into the synaptic cleft. The synaptic cleft stimulates the sarcolemma and makes an electrical charge. Okay, that electrical charge travels down the surface of that muscle cell, and as it goes down that muscle cell, um, it's going to create the contraction. Now, when you have that muscle cell, there's ATP at work here, and it's going to need to trigger those actin and myosin proteins to move against each other. Those are the muscle fibers. Okay. So, what happens in a nutshell is calcium is stored in the cell. When there is stimulation, the calcium comes out of the cell and it causes that actin and myosin. It forms a bridge between them and they move. Okay. Then, as that stimulation on the sarcolemma goes past, uh, the calcium goes back into storage, and the actin and the myosin stop moving. They are no longer moving, and they go back to a resting state. Let's look at what happens. Okay, so you have a resting cell. The resting cell has a lot of sodium on the outside of it. There's potassium, and potassium is inside the cell. Now, the positive charge on the sodium is higher than the potassium. Potassium is able to move in and out of that cell. It leaks out. Okay, it's on the inside, but it's leaking out. And that means that it's more positive charge on the outside of the cell and it's negative on the inside of the cell. That is called polarization, and it has action potential. Okay, you have positive one pole on the outside, kind of like a battery, right? Battery has a positive end and a negative end. So you have positive outside of the cell and negative inside the cell. And that's polarization. When that acetylcholine is released from the neuron, the sarcolemma opens sodium gates, and that sodium goes rushing into the cell. And it now creates positive on the outside and a positive on the inside. So they're the same, and that's called depolarization. When you have depolarization, calcium comes out of its storage, forms a bridge between the actin and the myosin, and they move against each other, and that is muscle contraction. Okay? Then, as the stimulus goes by, the calcium returns to storage, and ATP starts pumping that sodium out of the cell. It cannot diffuse, right? Because it's still positive outside the cell, it's positive inside the cell. It's just going to sit where it is. But we have to return that cell to a resting state. So, what happens is ATP starts pumping that sodium out of the cell, and it once again establishes repolarization. The inside of the cell becomes negative again. The outside of the cell is now positive, and that is ready. The cell is now resting, and it's ready for a new stimulus from a neurotransmitter. Here is a diagram of what's going on. Okay, don't worry about T-tubules. Okay, you don't need to know what those are, but you just need to know that you can see the nerve cell, the axon terminal of the motor neuron, is dumping acetylcholine into the synaptic cleft, and that acetylcholine is acting on that sarcolemma. Okay, and where those motor neurons hit the sarcolemma, there's like a little divot that they sit in. Okay, it's not flat. There's a little divot that they sit on, and that's what starts this electrical charge. Okay. Okay, here we go. So you can see here, there's action potential. All right. Axon terminal. The nucleus you can see over here. There's a nucleus over here. You can see all the different sarcomeres. This is a myofibril. You can see those. This is a single muscle cell. Okay, so um, from excitation to relaxation, action potential. There's an electric signal that travels down the nerve cell, causing it to release the acetylcholine into that small gap. Okay, the synaptic cleft. The neurotransmitter crosses that gap and binds to the muscle cell membrane and causes action potential in that muscle cell. When that action potential travels down the sarcolemma, the sodium gates open and the sodium floods into the cell, creating a positive area outside the cell and a positive area inside the cell. That's depolarization. When you have depolarization, the calcium ions are released from storage. They flow into the cytoplasm, and the actin and myosin forms a bridge between those actin and myosin filaments, and they move against each other. Okay. And that's what causes the contraction. Once that action potential is passed, the calcium gates close, the calcium goes back into storage. The sodium gets pumped out of the cell and it returns to a polarized state, which is the resting state. And now you have a cell with resting potential. Okay. We will be going over this in class, and we will be doing a little little game that I made to make it easier for everybody to understand it. So that's all I'm going to say about muscle contraction. I went over a little bit longer than I wanted, but it's really important that you guys understand this, and we'll talk about it more in class.